Hi my dear students, welcome back to Tungal online classes. In the last class, we already discussed regarding gel electrophoresis. The gel electrophoresis is the technique which is useful for separation and isolation of DNA fragments from agar-agar medium. And also I give the information regarding cloning vector characters. Cloning vector, it should be containing three characters, like one is worry site, second one is selectable markers, and third one is cloning site. And also we discussed regarding PBR 322 plasmid also. The today class is how to selection of this recombinant bacteria or cells from non-recombinant bacteria. The selection process is done with the help of selectable marker genes. Selectable marker genes. Here, generally some of the bacteria, the code for resistant to antibiotics, such are like tetracyclines, ampicillin, and kanamycin. Clear? Let's write how this is useful for selection of recombinant plasmid. First point, for selection purpose, with the help of selectable marker genes, is used for selection process, and normally some bacteria are code to resistance to antibiotics, such are ampicillin, tetracycline, and kanamycin. So these three are one type of antibiotics which have the capacity to kill the bacteria. But by using of these three antibiotics also, some of the bacteria do not going to die because they consist resistant power to these antibiotics. So that's why they are going to survive. So such a genes we are going to using as selectable markers. So here such a resistant genes, are used as marker genes for selection of recombinant cell. For selection of recombinant cells. Here already PBR 322 plasmid is here. Two marker genes. Here these genes for resistant to tetracycline. These genes for resistant to ampicillin. Here two resistance genes we are used for preparation of PBR 322 plasmid. What they are? One is AMPR. It means ampicillin resistant genes. Next one is TER, tetracycline resistant genes, or means resistant genes. Now these two genes are which are used for selection of recombinant plasmid. Let's see how it is useful, how it is going to work, out. For that, what you have to do, just ligate the desired gene at a recognition site of any one of the two resistant genes. For example, BAM1, it is a recognition site. In that site, I am going to ligate the desired gene. Where is the BAM1 is present? The BAM1, it is present in within the resistant site of tetracycline. Resistant site of tetracycline. Simple. Then what happen? If you insert this desired gene in this tetracycline resistant site, it is going to lose its character. Okay. Then only one marker gene is there, which that one is ampicillin. So now this PBR322 plasmid, due to the insertion of desired gene, it is lost tetracycline resistant gene character, but it is containing what? Ampicillin resistant character is there. So such is useful for selection of recombinant plasmid or recombinant cell. Let's see. Let's try the points. First, we have to ligate the desired gene at any one of the marker gene at a recognition at a resistant sites. Resistant site. We have two sites is there. Out of two, any one of the site we have to use to ligation of DNA. Then second, what happened? Due to this ligation, it going to lose the tetracycline resistant capacity. Lost the tetracycline resistant capacity, but it is already containing, but it has the capable for resistance to which one? Ampicillin to ampicillin. Okay. Now where we ligate the DNA? Which site? Ligate the desired gene at a BAM H1 site. In BAM H1 site, we ligated the DNA. Then see here. Now this cell, this recombinant cell, we have to plate it on two medias. Let's take two medias. So this is equal to one Petric plate, and here also be one Petric plate. Okay. So what have to do? So first we have to add the medium. Here we added the medium. You know what is medium? The medium is a combination of micro, macro and essential element, which is also containing growth factors. Okay. Next. Now in this medium we added which antibiotic? Ampicillin. Antibiotics are added. And here one more medium is prepared. So this is dish. Petric plate we have to call. Petric plate, nothing but one dish we taken. In this again medium is present. Now this medium we added a tetracycline antibiotic is we added. Now what have to do? Simple. Recombinant plasmid containing E. coli bacteria, first we have to add on ampicillin medium. So this red mark. So this is what? A recombinant plasmid containing E. coli cell. Recombinant plasmid containing E. coli cell. Okay. Then what happened? Here, this recombinant plasmid containing E. coli cell, it is already resistant for ampicillin. Ampicillin genes are there. Then it going to resistant for ampicillin. So it is not going to affect the bacteria. So it will grow. It grow on the medium. After some more time, it is going to get double. It means replicated. Going to replicated. After some more time happen on the media, we can notice number of E. coli cells. E. coli cells. Got the point or not? Next, these cells are transformed to in this medium. Now these cells are which cells? Recombinant plasmid containing E. coli cells were transferred from one medium to another medium. Now this medium is containing what? A tetracycline containing medium. Now what happened? Here we added. So after some more time we notice that this E. coli bacteria, it is not going to grow, not replicated. After some more time it is going to die. Why? Because why it is die? Why it is not grow on this medium? That is actual question. Why this bacteria survive in only this medium? Why not on this medium? The answer is what? The answer is, sir, already this plasmid is resistant capacity to ampicillin. So that's why it is surviving in ampicillin medium. And in this case what happened? Here tetracycline antibiotic is there. Sir, we already insert the desired gene at a tetracycline site, so that's why it is unable to produce the resistant power to tetracycline. So due to that, this bacteria is going to die. Got the point or not? So by this process, by simultaneously plating, by using of two plates, Patrick plate, we can select the recombinant plasmid containing E. coli cells. Got the point or not? Just write it. The first point, a recombinant plasmid or recombinant cell, you have to write directly. A recombinant cell, E. coli cell, it grow only on which medium? Ampicillin medium. Ampicillin medium. Why? Because it is already containing ampicillin resistant genes. It is here replicate. Okay. Next, here these are transform to another medium, that is containing what? Tetracycline medium. Tetracycline medium. In this tetracycline medium, the recombinant plasmid is not grow, it is die. Why? Because we already added desired genes in this tetracycline region, so it is not going to code for resistant power to the tetracycline. So due to that, what happened? It is die. Okay. So in this process, by this process, we can select transformed cells, transformed or nothing but recombinant cells, okay. But in NCERT clearly it is given, so it is a cumbersome procedure. Cumbersome means it is clumsy, little bit not clear method. So further, the scientists have already developed one more technique, alternative for selectable marker genes. In the first case, we used two Petri plates were used. Simultaneously, we have transferred the recombinant cell from one medium to another medium. But in the NCERT book, it is clearly given, it is cumbersome procedure, it is little bit tough to understand as well as clearless concept. So that's why they developed one more technique. It is an alternative technique for selectable marker genes for selection of recombinant cell. Here, for selection of recombinant cell, one alternative technique is there. They developed one alternative technique for selection of a recombinant cell. So the technique is nothing but insertion inactivation, insertional inactivation, it is a technique. So in this case here the technique is differentiate a recombinant to non-recombinant by ability to produce color in the chromogen reagent. Got the point or not? One alternative technique is there. The alternative technique is nothing but insertion inactivation. It is which is useful for differentiation of recombinant cells from non-recombinant cells by the ability to produce the colors in the presence of chromogen chemical or chromogen reagent. For that, what have to do? It is very simple and clear concept. Here just we have to insert the desired gene at a Z gene location which is already present in PBR 322 plasmid. This Z gene have the capacity to code for the beta galactose enzyme. So now what we are doing, inserted at a Z gene location. Then what happened? The Z gene is inactivated. Now it is no capacity to produce beta galactose enzyme. Okay, that is called as what? Insertion inactivation. It is a 2-mark question. Insertion inactivation. Let's write the point. Here, desired gene, no need to get confused. Desired gene, alien gene, foreign gene, wanted gene, these all are same. Okay, desired gene is ligated at a which site? Z gene site, which is already present in PB or 322 plasmid. Now what is the function of Z gene? The Z gene is code for one enzyme. Enzyme is nothing but what? Protein. Code for one protein, nothing but enzyme, that is beta galactose enzyme. Beta galactose enzyme. Now due to insertion, what happened? So these Z genes are inactivated and they have no capacity to produce beta galactose enzyme. So this process is called as what? Very important 2-mark question. Insertion inactivation. It is called as insertion inactivation. It is called as insertion inactivation. So this technique is useful for recognition of recombinant cells. Let's see how it is going to use for. So same for that, I have to use medium. Same, this is one Petri plate. Again add medium, but this time we are not adding of ampicillin as well as tetracycline. This time we are adding one chemical reagent, that is chromogen. Chemical we are adding. Okay, so this is a chromogen chemical. And now we added, this is E. coli cells. Which cells? Insertion of E. coli cell. Insertion E. coli cell means what? It is containing desired gene. Okay, then what will happen? Let's check is, if this medium is containing normal cell, it means if the medium is containing other than non-transformed cells, what happen? If the medium is containing transformed cells, how it is going to react with chromogen? Let's check it. First, a recombinant cell, nothing but what? Insertional cell. Maybe for example, sometime other than recombinant cells also growing. Here we can call as non-recombinant cells. Now what happened? Just here these two cells are going to react with a chromogen. Here also going to react with a chromogen. Then what happened? Here this cell is insertional cell. It is have no capacity to produce beta galactose enzyme. Then it is not going to react with the chromogen chemical, so it is not going to produce any color. The bacteria colors is colorless. These bacteria do not perform any color. Now in this case, non-recombinant cells are maybe grow on the medium. So the non-recombinant cells, nothing but bacterial cell, it is also containing Z gene that code for beta galactose enzyme. Then they are going to react with the chromogen chemical and they become as blue colored. They become as what? Blue colored. Clear? So here, if the bacteria are formed blue color on medium. If they are blue colored on the medium, they are what? Non-transformed cell. These are non-transformed cell. Nothing but non-recombinant cell, just equal to pollutant. Okay, then which are colorless? Such are what? A recombinant. Which are recombinant E. coli cells. So this is very easy process to selection of recombinant cells from non-recombinant cells. Okay, so that's why at the beginning, the principle of genetic engineering, first one is RDNA technology, second one is maintenance of sterile medium in ambience. So beside the medium, the area should be what? Non-contaminated area. Microbes should not be present. Clear? If may be present, we are going to use this method. Clear? Now what we did, with the help of restriction endonuclease enzyme, we cut the plasmid, as well as same restriction enzyme is used for cutting of desired DNA. Okay. Next, In the next step, we prepared artificial plasmid, suitable vector we prepared. Okay. Then later, with the help of restriction, with the help of ligase enzyme, desired gene is added to the plasmid and become as recombinant plasmid. That is entered into the E. coli cells. That E. coli cell is growing on the medium. On the medium, by using of insertion inactivation technique, we are selected the only recombinant cells from non-recombinant cells. So this is happened. Let's try to find what we did. First, by using of a restriction endonuclease enzyme, we are cut the plasmids. We are cut the plasmids and as well as desired gene. Same enzyme we have to use. For example, BAM H1 is used for cutting of plasmids as well as desired gene. Which plasmid were used? PBR 322 plasmid were used. First step is finished. And second step, we prepared a cloning vector. Cloning vector is prepared. Next, after preparation of cloning vector, by using of ligase enzyme, we are ligate the gene, desired gene into plasmid. Now it is become as what? R plasmid. And now this is we are entered into the E. coli cells. Now these E. coli cells we can call as what? A recombinant cell. It is called as recombinant cells. Now these recombinant cells will transfer on medium. In medium what happen? It is going to replicate. To form of number of cells are formed. And later by using of insertion inactivation. By using of insertion inactivation technique, we are select the recombinant plasmid containing E. coli cells from non-recombinant cells. It is selection of R cells. So this is actually we already did. Next information is regarding cloning vectors for plants as well as animals. It is also one important topic in NCERT. See here, Agrobacterium tumefaciens, it is a bacteria which have the capacity to cause the disease to the dicot plants. It causes the disease to dicot plants. Next, cloning vector for plants as well as animals. Here, for example, one bacteria is there. The bacteria name is Agrobacterium tumefaciens. It's a bacteria. It is a pathogen to dicot plants. It causes a disease to the dicot plant. The disease is here tumors. It can induce the tumors in dicot plants. How it is going to perform this? Simple. When the bacteria is entered into the eukaryotic cell of plant cell, it going to release one small DNA fragment that is called as tDNA. That is called as what? tDNA. So this tDNA is going to transform, it means change the normal plant cells into tumor cells. Now these tumor cells are going to produce some chemicals which is needed to the bacteria. That chemicals which is required to bacteria. So here what we have to focus here, this Agrobacterium tumefaciens bacteria, it has the capacity to transform the cell. It has the capacity to transform the cell. Let's write. The heading is cloning vector for plants as well as animal cells. Simple. For example, we have taken one bacteria. The bacteria name is what? Agrobacterium tumefaciens. Agrobacterium tumefaciens, it is a bacteria. It is a pathogen to dicot bacteria. Pathogen means disease causative agent. Here, due to that, it causes the disease is tumor. It is cause a disease what? Tumor. Here when this Agrobacterium tumefaciens, when it is enter into plant cell, when it is entered into the plant cell, it is going to deliver one small fragment of DNA that is called as tDNA. It is called as what? tDNA. tDNA. It means it induces tumors in dicot plants. Okay. Next, this tDNA, which has the capacity to transfer the normal plant cells into tumor cells. It has the capacity to transform normal plant cells into tumor cells. Now each tumor is containing tDNA. Then what happens? This each cell is going to produce some chemicals. So these chemicals are actually need to Agrobacterium. Clear? Here what you have to notice here, these bacteria have the capacity to transform the normal cells into tumor cells. Okay, same like as for example in human retroviruses, same. So these also have the capacity to transform normal human cells into tumor cells. Now here, by using of biotechnology, these two virus as well as bacteria, we are prepared for vectors. Now these two retrovirus as well as Agrobacterium tumefaciens, these two we are using as a vectors, clones. So before that, what we have to do? Before that, this tDNA, in this tDNA, so which is have the capacity to cause a disease to the plant. Such genes are deleted. It means harmful genes eliminated from tDNA. So now this agrobacterium containing tDNA, it is not harmful to the plant, but it can perform, but it have capability to perform transformation. Got the point or not? Next, after deletion of these harmful genes in the bacteria, in such location where inserted desired genes. Desired genes are inserted in such location. Now what happened? Now these already have the capacity to transformation. Now what happened? Now normal plant cells is become as desired character containing gene cells. Normal plant cells are become recombinant cells, and each cell is containing desired gene. The same process also we are using in here also, retroviruses also. Same in retroviruses, first we are going to cut or elimination of harmful genes. Harmful genes are eliminated. Now this is no harmful to, it is not cause a cancer to the human, not harmful. Okay, it is called as disarmed. It is called as disarmed cell. It is called as disarmed cells. Now same in such location again wanted genes are inserted. Now these retroviruses again perform transformation. Okay, got the point? So these are cloning vectors. It means Agrobacterium tumefaciens bacteria, we can use the cloning vector for plants, and retroviruses containing genetic information, which is also using for cloning vectors in animals. Okay. Next, competent to host cell. Here competent means here it is a ability to receive the plasmids or DNA from outside to inner side of the cell. So that is actually competent to the host. Got the point? Here, for example, we prepared one recombinant DNA is prepared, and here E. coli bacteria is ready. Then if you kept beside of plasmid as well as E. coli cell, this E. coli cell is not going to receive the plasmid from outside of the environment to inside of the E. coli cell. That is actually a problem. So to overcome this problem, we have to give the treatment to host cell. Then only it is receive the DNA from outer side to inner side of the cell. Got my point? Let's write. Competent to host cell, it is nothing but it is the ability to receive DNA or plasmid. Which plasmid? Recombinant plasmid from outside the cell. From outside the cell. Clear? First, here this DNA or plasmid, DNA or nothing but plasmid, both are same. So this DNA generally it is hydrophilic nature. Hydrophilic nature. Due to the hydrophilic nature, it can't move or it can't across the cell membrane easily. So it is not move or across the cell. The reason is here why. Sir, why it is not possible to across the cell membrane? So for that you have to refer in the 11th class fluid mosaic model we had been discussed. Just check it. Then tomorrow you have to give the answer for that. Here, so here for that we have to give the treatment to the cell. Treatment to host cell. Treatment to the host cell. So the treatment is generally we have to treat with the cations, divalent cations. We have to treat with the divalent cations like calcium. When we are going to treat the cell with the divalent cations as well as calcium, it's going to lead small gaps on bacteria cell wall. So this small gap is enough to movement of plasmid or DNA from outer side to inner side of the cell. Through this small gap, our plasmid or nothing but DNA, it enter into host cell. Later it is going to self-replicated, that you know that already. So this is actually first method. And second, one more method is there to complete the cell. One more method is simple, the placing of alternative treatment of heat or alternative treatment of chilling treatment as well as heat treatment. Simple, take a one beaker. The beaker should be consist of recombinant plasmid as well as one E. coli cell. Just place on ice for some more time and later give the treatment at least 42 degrees Celsius heat shock you have to give. Then later immediately that put them on ice. By this alternative ice chilling treatment and heat shock at least 42 degrees Celsius. So after some more time the bacterial cell wall is going to little bit loose. It's going to give small tunnels, small gaps are formed. So through this gap again the plasmid it can enter into the bacteria. Let's write the points. Here plasmid, next host cell. We have to treatment like alternative treatments of ice. First you have to place on ice for some more time for chilling purpose. Later immediately give the heat shock treatment at 42 degrees Celsius, not more than, because if it is more temperature it is going to harmful to bacteria as well as DNA. Then immediately again put them on ice. Then what happened? So due to that bacteria cell wall become loose and small gaps are formed. So through these gaps, so this plasmid or desired DNA it enter into E. coli cell. E. coli cell. Clear? Next, vectorless transfer. Without using of vectors also we can transfer the recombinant material into the host cell. It is vectorless transfer of genetic material into the host cell. It is possible or not? Yes, it is possible. Recent techniques. Just put the heading vectorless transfer of a recombinant DNA into host cell. Okay, this is actually done. Here first, by using of micro injection one technique. Second, biolistics, nothing more we can call as a bombers. Third, electroporations. Fourth, chemicals. And fifth one is disarmed pathogens. So these 5 techniques we are using the transfer of genetic information by without using of vectors. Right, first one, very simple and easy one, micro injections. So here a recombinant DNA directly injected into the host cell to near to the nucleus. Here a recombinant DNA is directly injected to host cell. So this method especially used for animal cell. By using of micro injection we can insert the genetic information in animal cell. First one. And the second technique is bombers, nothing but biolistics. So this is for plant cells. Why? Because the plant cell is containing cell wall, we have to break the cell wall first. So further here bombers, biolistics, it is also called as gene gun. It is also called as what? Gene guns are used. So here the first we have to prepare one bullet. The bullet is nothing but recombinant DNA. So this recombinant DNA is coated by gold or tungsten. Now this is what become as bullet. Now this bullet is inserted in gene guns. By forcibly we are breaking the cell wall of plant and it is entered to near to the nucleus. Okay. Next, here the DNA is coated by gold or tungsten chemical. Now it is become as what? One bullet is formed. So this is inserted in gene guns. By forcibly we are going to enter into the plant cells by some speed. By you can see high velocity we are going to break the cell wall and it is entered into the nucleus. Okay, so this is second method, bombers or gene guns. And third, one more technique is there here, poration. Electrical poration. It means making of pore on bacterial cell wall or plant cell wall by using of electrical current. By using of electrical current we make small pores on bacteria cell or plant cell. So the pore size is according to the DNA size. DNA size is small, we are going to make a small pore. If it is big size, we are going to little bit big pore we are going to make with the help of electrical current. Next, fourth one is by using of chemicals. By using of chemicals also. Here PEG chemical. PEG chemical. It means polyethylene glycol. Polyethylene glycol. It is a chemical. By using of this chemical, when using this chemical, the host cell wall it is going to cleave, going to form small gaps are going to form. Through these gaps, the desired DNA enter into host cell. This is one more technique. And the last technique is disarmed pathogens. Disarmed pathogens. What are the disarmed pathogens? We already used it. Which one is? Agrobacterium tumefaciens plasmid, nothing but a tDNA, as well as retroviruses. Disarmed pathogens means what? But disarmed, diseased causes to genes are eliminated. Now the pathogen is no more harmful to plants as well as animals. So there are such type of are called as such type of genes we are using for cloning. Write it. Example or what? We already discussed. One is TI plasmid, tumor inducing plasmid. So disarmed plasmid which is used for plants. And next one is a retrovirus. Retrovirus which is used for animal cell. So this is today class. In the next class we are going to discuss regarding process of recombination DNA technology. Thank you all of you.